In the last several years, Joel started looking into the game between cancer cells and other molecules, and he's using different mechanisms of ecology and evolution to understand, to define, and to treat cancer. And we asked him to give a talk on this topic today. I just want to remind you that Joel published some 250 papers with 16,000 and more citations, a lot of other contributions to science, and it's a great pleasure to have him here, and thank you very much for you willing to give us 2 talks on one day. Please welcome Joel Braun.
Yaron, thank you very, very much. Yaron didn't mention, but we are academic SIBs. So that's very, very cool to sort of be here with my academic brother. Nope, not a worry. And I thank you all for being here. To come for 2 talks, especially after such a wonderful day. The talks have been fantastic. The people I've had a chance to talk to have been amazing. And quite personally, I don't think I would come to a second talk except I have to give it. So I really want to thank all of you for very kindly being here and try to explain then how many of the principles and ideas we've been talking about here at this conference actually apply quite closely then to cancer. So I'm an evolutionary ecologist, and I would describe that as somebody who studies ecological systems through the adaptations of their organisms. The other feature you can discover is in my lab, we've worked with diverse organisms, and it's like many things. How many of you, of course, these are the people in here that have been around the block a few times, suddenly realize your life only makes sense in hindsight, and you discover things about your life you didn't know. And one of the things, this would have been obvious to me, but in fact, throughout my career, I never actually really chose my study organisms. In fact, my colleagues, my friends, my graduate students, and my postdocs have always chosen my study organisms, and I guess cancer will be no different. My ties here to Israel go very, very deep, thanks to Bert Kotler. Yes, this is 1986. This is at Bet Guvrin there. Bert and I were younger there then, so was your own, actually. And I don't remember the days, but those are the days at Bet Guvrin where they didn't actually have any kind of facilities. You could simply go there with a headlamp, and you could spend any time you liked going down these rabbit holes of these sort of ancient Bar Kokhba sort of era caves and whatnot. And of course, now I work with Bert Kotler. We have a large aviary where we're able to study predator-prey interactions. And some of this will actually come up in the talk. And this is down at Stable Care where we're actually currently doing research. But before I dive into things, we're going to be talking about cancer. How did I actually wind up in cancer? It was Tom Vincent. I've always been interested in game theory as the modeling framework that I like to apply then to evolutionary ecology. Tom Vincent then was my game theory mentor. He was an aerospace and mechanical engineer at the University of Arizona, and we worked together for several decades, many, many years, and he began working with a gentleman, Bob Gatenby, who at that time was department head of radiology at the University of Arizona Medical Center. And Bob Gatenby is very much a Renaissance thinker and was wondering whether one could apply evolutionary principles to cancer. In particular, could you use game theory? And I'm putting up here, these are all the papers that Tom Vincent and Bob Gatenby wrote together, and you'll see many of them applying game theory. A gentleman named Tomlinson is probably the very first person and paper in the late 1990s to apply game theory to the notion of cancer, but it's really Bob Gatenby and Tom Vincent that ran with it. Now, throughout this period that they were publishing these papers, Tom Vincent just kept imploring me, you must work on cancer. You see this trend of sort of selecting things. And anybody that knows me is I tend to spread myself way too thin. I find myself being a kid in a candy shop when it comes to science, and so while there was this urge to say yes, there was this realization that if I did, I would let them down there. And so anyway, so I kind of said, look, I'm happy to read the paper, this is all very interesting, but Bob persisted and persisted. And so finally, in Wrocław, Poland, June 2008, at a conference on dynamical game theory, I said yes. I am happy to join you. It's Tom Vincent, Bob Gatenby, and myself, and indeed we did outline our first paper on a napkin. That is how this paper came across. And then 1 month later, Tom Vincent phoned me up and said, I have terminal pancreatic cancer. Never was I so happy that I said yes. And so at that time, Bob Gatenby had moved to the Moffitt Cancer Center, where he established a number of integrated programs to try to approach cancer from diverse elements. And one of the programs that he started, and is headed by Sandy Anderson then, is right now the Moffitt Cancer Center is the only cancer center in the world that actually has a department. An entire department devoted to integrated mathematical oncology. There's a few other institutes and now, which is good, that are hoping to be able to replicate perhaps the significance of having mathematical oncology, but this is to show you just how many there are of us at Moffitt that do mathematical oncology. There's 6 of us that are faculty members. There's a large number of graduate students, postdocs, and indeed, a number of these come from an ecology and evolutionary background. So we're really a very, very diverse background. That's kind of the goal. In today's talk, much of the work that I'm going to be presenting to you is going to come from Jun-Song Zhang. He's the head of prostate oncology at Moffitt, and he is going to be the lead person on a clinical trial that I'll show you later in the talk. See Bob Gatenby, Jessica Cunningham has been kind of this wonderful collaborator, and now she's a graduate student who's been involved in a lot of the modeling. And then Mark Lloyd was my first graduate student in cancer biology. He's fantastic as an imaging analysis analyst, and he's now part of a startup company that he created, Inspirata, that's there in Tampa. So now cancer is interesting. It was in the early 1970s, Richard Nixon declared a full-court press to try to cure cancer. He didn't use the term, but very quickly it became known as the war on cancer. So the war on cancer gets declared in the early 1970s. And the idea then was if we could have a concerted effort, we would be able to cure or deal with this scourge. Now what's interesting, if we look back now almost 50 years, we now have about every imaginable I thing. We put people on the moon. We have bullet trains. But in terms of cancer, I will suggest to you that while there have been notable successes in detection, early detection, there's been huge successes in being able to cure stage 1, stage 2 levels of the disease. Once cancer is metastatic, it will kill you. There's a few exceptions, that's good, but it will kill you. Furthermore, 1 in 4 of you, look around the room, will likely die of this disease. You will die of cancer. And so the cure rate, even though we can prolong life, we can do things, the cure rate when it comes to metastatic cancer has barely budged. And somebody finally gave me the statistic, they were looking it up, and that is over $500 billion later has been spent to try to deal with this particular problem. So while there has been improvement, I think almost all of us would agree, it's been very incremental, and it's been very, very expensive. Then our vice president under Obama, Biden, his son died of cancer, and so Obama and Biden, they declared the cancer moonshot. And the idea here was if we could put a man on the moon, certainly if we devoted the same amount of effort and thoughts, we ought to be able to then deal with this particular disease. But I'm going to put to you that there's a big difference. When we were attempting to put somebody on the moon, we understood the physics. There was no disagreement over what needed to be done. There was no disagreement over the mathematics, the propulsions, the other type of systems. The real issue was, could we engineer a level of tolerance that would miss the moon? Could we bring them back? And could we ensure that the cell phone signal wouldn't drop unexpectedly at a really bad time? On the other hand, I'm going to suggest to you that with cancer, we haven't even decided what the disease is. So in some ways, I think we've done extraordinarily well for a disease that we may not yet even agree on what cancer is. So I'm going to give you a moment to reflect in your minds what you think cancer is. So in your mind, you can write it down, whatever. In your mind, ask yourself, what would you tell somebody if you had to give a 1 or 2 line definition of cancer? Seems like we ought to be able to do that. So here it is. Cancer is one of the favorites, a disease of the genes, a disease of unregulated proliferation. As ecologists, I suppose we could call rabbits a disease of unregulated proliferation. And then there's the definition that you'll find on the National Cancer Institute website in the United States. Cancer is a group of diseases involving abnormal cell growth with the potential to invade or spread to other parts of the body. To me, that's about as exciting as describing a table as a horizontal plank, usually made of wood, sometimes something else, supported by some uprights. I'm going to kind of suggest to you that in the worldview that I take in the approach to cancer, is that actually cancer is a disease of Darwinian evolution. And in fact, the first principles of ecology and evolution are in fact the underlying principles that describe what cancer does. So there's the hallmarks of cancer. Hanahan and Weinberg, they published two versions. They had a shorter version, and then they eventually published this version. These two papers alone have been cited close to 60,000 times. Together, 60,000 times. Now, it looks great, right? A color wheel. I'm sorry for the 9% of the men in here who are colorblind. It doesn't look quite so great. But for the rest of us, it looks like a nice color wheel. These are the hallmarks. But in fact, I think what they obscure then is what cancer is and how they come into play. So here's how I'm going to unwind them. They don't look nearly as pretty. But the key message, if there's any message or thing that I get home this evening, is your take-home message, cancer is a speciation event. Cancer is where you, as a multicellular organism, spin off a new single-cell life form. It came from you, but it is not you. And whereas it was once part of the whole organism program, you, it now is the program. It has become the unit of selection. And I think what we can see if we examine and unwind kind of the hallmarks of cancer, resisting programmed cell death, evading anti-growth signals, enabled reproductive mortality, these are three features that are simply necessary for an organism to be the unit of selection. They're not really adaptations in themselves, they are just simply the properties of a level of selection that can now be subjected to evolution by natural selection. Then there's a variety they refer to here, deregulated cell metabolism. I think it's an unfortunate way to put it. Deregulated simply means the cancer cell is no longer doing what it used to do. But if the cancer cell is now the unit of selection, it makes no sense for the cancer cell to continue to contribute to the public good of the whole organism. So what it really should read is cease to perform whole organism functions. And then genetic instability. The last time cancer was a single-celled organism was probably at least 700 million years. The last time it was a single-cell species is 700 million years. How many of you agree that it has a lot of evolutionary catching up to do? So unsurprisingly, they actually evolve evolvability. As an adaptation, they evolve evolvability, and that becomes the serious problem because the failure of cancer therapy is indubitably that the cancer cells evolve resistance to the therapy. It is an evolutionary response. Sustained growth signaling is basically they gain independence. Now, if you look over here, these hallmarks of cancer are beginning to look like pretty sophisticated adaptations. Inducing new blood flow. Tumor-promoting inflammation brings in macrophages and other cells that will actually feed them. Avoiding the immune response. We would agree that safety is an important feature of all organisms. And then tissue invasion and metastasis means that as resources become limiting, the capacity to expand range and move into new territories and habitats. All of these represent basically ecosystem engineering. This all represents fairly sophisticated evolution. So like nature, cancer is a complex dynamical system. It's hierarchical from molecules to ecosystems, in this case, the tumor being its ecosystem. For today, what I'm most interested in, it is the product of evolution by natural selection. Nature can also be studied through the recipe of inheritance, the genetics. It can be studied as a historical process, phylogenetics, and the fit of form and function in terms of adaptation. What's interesting in the development of cancer as a field is to contrast it with that aardvark. If you look at that aardvark, much is known about its adaptations. The big ears, the long nose, the sticky tongue. We know a lot about its adaptations. We know some about its phylogeny. It's off there in Afrotheria. It's this kind of weird group. When in doubt, related to elephants. Always works. But we really don't know a whole lot about where it fits in. And finally, while we at Moffitt can do a whole genome sequencing in 20 minutes of somebody, nobody has ever done a whole genome sequence of even one aardvark. I've been tempted in our M2 Gen Center to slip in a blood sample of an aardvark, tell them it's a cancer patient, we need this sequenced immediately, and see, wow, that's cool. Would they even spot it? Would they even know what it is? On the other hand, if you now look at that hepatocyte, if you look at that liver cancer cell, we can do whole genome sequencing. We know the genetics up and down. In fact, personalized medicine right now for cancer is pretty much built around genetic signatures. Quite a bit can be done about its phylogeny, how it moves from the primary tumor to the metastatic sites. We know next to nothing about the adaptations of cancer cells. We know nothing about, we don't even know their generation time. We're not even sure what their limiting resources are. We don't even really know nutrient uptake. So my colleague Ron, who likes numbers and knows numbers, we are lacking so many numbers in cancer. So in taking an evolutionary approach from evolutionary ecology, what are Darwin's postulates? Heritable variation, like begets like with mistakes, struggle for existence, and then variation influences the struggle. So does cancer fit this bill? Well, cancer cells do evolve adaptations to their environment. Furthermore, cancer cells show considerable phenotypic plasticity. They can be quite variable in their response under different circumstances within the tumor. And so we can expect then cancers to first acclimate and then adapt to their circumstances. So if you see this possum in the picture, this is a possum in my backyard in Oak Park near Chicago. So many of you just might see a really ugly looking animal. How many people think it's cute? I think they're adorable. Oh, well, thank you. I love you guys. Possums, I think, are so cute. So what you might be saying is, wow, look at that possum, the only species of marsupial that is actually in North America. But what I see as an evolutionary ecologist is an ecological and evolutionary process running wild in the city, in my backyard. And it may not be a cheery thought, but I think it can be illuminating. That's the same thing. Cancer is an ecological and evolutionary process running rampant in you. Now, I think it relates to game theory. So what do we know about game theory? Game theory is the mathematics of trying to solve problems of conflict of interest. I think for our purposes, it's easiest to think about game theory as solving problems where my best strategy depends upon everybody else's strategy. My best strategy depends upon all of your strategies, and they may not align perfectly. So, does cancer fit the bill as being a game? As a game, the individual tumor cells are the players. Their heritable traits become their strategies. Per capita growth rates, proliferation and survival becomes their payoffs. The tumor tissue and environment sets the rules. And this is one that I think for oncologists, I think as ecologists, this isn't hard to handle, but for many cancer biologists, this is a tough one. And that is the game is primarily between the cancer cells. The cancer cells have no awareness of you. So I think there's this tendency, because we have such a big awareness of them, we somehow imagine they're aware of us. They're not. Primarily, the cancer cells are organisms in which you are the planet Earth, a very dynamic planet, and they are primarily trying to evolve at advantages, competitive advantages with respect to each other. But primarily, the important part is treatment strategies. As soon as treatment is imposed, treatment strategies then become part of the game. So how can we do this mathematically? And I won't run through too many details, but just to give you a flavor of what we do, we imagine there's heritable variation. Organisms possess strategies, and these heritable traits are part of some set of evolutionarily feasible traits. The struggle for existence says organisms have per capita growth rates. This can be represented here by X as population size, dx/dt is change in population size. And at some point, if the population size gets big enough, fitness declines with population size. There's just some sort of limits to growth. So the first question, of course, is how big is my strategy set? And of course, if you look at domestic dogs, you can imagine that what is evolutionarily feasible in a wolf is huge. How many of you can picture the inner chihuahua in a wolf? Better yet, how many wolves are embarrassed to acknowledge that the inner chihuahua is part of their set of evolutionarily feasible strategies? So we would agree that within domestic dogs, wolves, it's huge relative to what we see. Same thing with cancer cells. Cancer cells have the entire human genome to draw on as part of their evolutionarily feasible strategy set, plus all of the myriads of ways of combining epigenetics, methylations, chromosomal rearrangements, episomes. So when you begin to catalog, it's not surprising that cancer cells have a lot of evolutionary potential. Then there's the struggle for existence. Struggle for existence, first two laws of ecology. All populations have the capacity to grow exponentially under ideal conditions, but no population can grow exponentially forever. There are limits to growth. What is a really fascinating question, I'm quite interested in this one, that means there's limiting factors. As ecologists, we often know what is the limiting factor for our system. In cancer, we're not even really sure. For some people, they're convinced it must be glucose, others oxygen, some micronutrients, perhaps fatty acids, others nitrogen. Is there even nutrient cycling? We know that cancer cells, when they die, they seem to pick over each other, almost like vultures on a carcass. But we don't even know how many times a nitrogen atom goes around inside the tumor before it flows out of the tumor after having flowed in the tumor. So some of the most fundamental pieces of knowledge that we would expect to have for ecological systems, we have thus far not learned in cancer. And I would argue that knowing these could be quite useful. So how do we put it together? We create what's called a fitness generating function, and that just says my expected fitness. My expected fitness is a function of my strategy. I'm the focal individual, the strategies of my neighbors, and the population sizes of my neighbors. So we can put all of this together then to describe how evolution by natural selection might proceed then in cancer. And we use this as our tool then for modeling the population and frequency dynamics that goes on in cancers. And in fact, this is what I will posit. We'll go back to our domestic dogs. Cancer cells are likely a rather dull subset of what is evolutionarily feasible for them and what is ecologically acceptable. How many of you could picture, maybe nobody would want to watch it, how many do we have dog lovers in here? If you just plug your ears for just a moment, here's the new reality TV show. Tonight we will release the dogs of the Serengeti. We will start with 100 chihuahuas. We will release them as the sun is setting and come back tomorrow morning to see how they're doing. So if we were to release 100 chihuahuas out on the Serengeti, how do you suppose they'd do? How many people would watch that reality TV show? You are a sick human being. No, just kidding. I would watch it too. How could you not watch it on there? How many people think they would starve? Wow, you really have confidence in them. How many people think they will become chicken nuggets for all of the predators long before they can starve? How many think the chihuahuas will just run up to a water hole when they're thirsty rather than looking to see whether there's a crocodile at the water hole on there? So this experiment's been done throughout the world. We have pitted the great variety that we know of as domestic dogs, and they essentially all come out looking like dingoes. There can be ecotypic variation, whether these are wild dogs in the Smoky Mountains in the United States, whether these are dingoes in Australia or wild dogs elsewhere. Furthermore, we can outline then the evolutionary dynamics. The ecological dynamics are simply described by G multiplied by population size. The strategy dynamics is simply this property that the average strategy of this group of cancer cells will change according to the fitness gradient. This is just having some sort of adaptive landscape. And K here represents an evolutionary speed term. What's interesting in cancer is that ordinarily this speed term, because the way the cancer cells can evolve epigenetically, the ways that they can evolve via chromosomal inversions, means that in general in ecological systems we assume that the ecological dynamics are much faster than the evolutionary dynamics. What's a fascinating feature of cancer is that may not be so. But what we do know is in evolutionary systems, we can expect systems in ecological and evolutionary systems to go to ESSs, a strategy which cannot be invaded by alternative strategies when it is common in the population. Plus, there may be a variety, one or more species that can be present at the ESS. Here's what I think is one of the biggest questions for me in cancer biology. This gets me excited, and part of the research of my lab is devoted to this question, and that is, does tumor evolution progress towards niche filling and niche partitioning? Can we find the Darwin finches of a tumor? And if so, are most tumor cells most of the time at or near their ESS? So here's an adaptive landscape with 5 species, all of them nicely occupying peaks on their adaptive landscape. Right now, this question is unresolved in cancer. The general models you'll see again and again and again, I refer to as spilt milk models. There's a sort of starting group of cancer cells. We know they develop a myriad of mutations. You find huge genetic variation. They call it microenvironmental heterogeneity. And you find a huge amount, and then suddenly, bam, we do some sort of therapy. This therapy then, there's only a few of these individuals that are able to survive the therapy. There's a bottleneck, and then this process of the spilt milk begins again. And so right now, there's considerable interest, but as well as, is this the appropriate model for approaching the diversity of different cell lines within a tumor, or is this approach? And to give you some sort of examples of how this works, I'm going to start with my 2 favorite species out there. One of these is the fox squirrel and the gray squirrel. Both of these are in my backyard in Chicago. And what you have then is the fox squirrel is not a particularly good competitor, but it's very tenacious with predators. The gray squirrel down here, smaller in size, very sophisticated when it comes to competing for resources, but very vulnerable to predation risk. So in fact, gray squirrels in general tend to live in the deep woods, where they're safer, there's more food. Fox squirrels tend to exist on the wood margins, where there tends to be more conspicuous to predators, less competition for food. So unsurprisingly, food safety trade-offs are one of the primary drivers of biodiversity, say, within a local area. So we asked the question, can we find fox squirrels and gray squirrels in a tumor? There's good reasons to expect them to exist. And again, we went looking for them by predicting them in advance. We did not collect the data and then try to interpret the data. And so what we did is we took more of an ecological approach. So what we did, here's a breast cancer histology. We then laid down transects. Typically, they will do the whole histology sample and they will count up the various cell types. What we did is we then would throw down 3 transects in the interior of the tumor, 3 transects at the edge of the tumor. And we did this for 15 women, stage 1, stage 2, stage 3 breast cancer. The idea is that along the edge, there's going to be more nutrients. It's less nutrient limited because you have the normal cells and the normal blood flow, but you're also exposed to the immune system. On the other hand, deep here in the interior of the tumor, there's going to be less exposure to the immune system. It will be safer, but it will be far more competitive. So let's see if anybody's listening. Which species of squirrel do we expect to find on the edge of the tumor? Fox squirrels. Who do we expect to find in the interior? Gray squirrels. Now, the trick was to sit down with cancer biologists and define what are the adaptations that would represent food and safety. 2 that came to mind is what's called CA9, CA12. These are carbohydrases. CA9 and CA12 are known as biomarkers. Both of them can be upregulated in tumors, but nobody had asked the question, do all the tumor cells upregulate CA9 and CA12, or do some do one or the other? CA12 we identified as a gray squirrel strategy. The CA12 is a buffering agent. The CA12 tends to engineer your environment. And the CA12, while it may enhance your exposure to the immune system or your lethality of the immune system, in fact makes you a better competitor for resources. CA9, on the other hand, is an insidious adaptation. It basically creates a resistance to acid, so the cell then produces an acidic environment through glycolysis, and then CA9 is what allows it to live in it. But that CA9, between doing that acidic environment, among other things, creates an acidic moat between the cancer cell and the killer T cells. The T cells must make contact to destroy the cancer cell, and they cannot cross this acidic moat. So we asked the question, how are these upregulated in a tumor? And the results, I have to confess, were more astonishing than I was even expecting. So what you have here is all these yellow ones. They're upregulating CA9. All the blue ones are upregulating CA12. Almost no cancer cell in any of the 15 women were upregulating both. They were either on the edge of the tumor, upregulating CA9, or they were in the interior of the tumor, upregulating CA12. This is what the data looked like. It's almost statistics free. You can look at the edge, and in the middle, you can see the CA9, the CA12. It doesn't matter which grade of cancer in these women. I think you would agree that the results look pretty good. Most of us, if we were doing a survey of habitat selection in animals, your own would be pretty happy with these results. It suggests a couple of things. Yes, it looks like tumor cells then, at least in breast cancer, there are both fox squirrels and gray squirrels. They have ecological attributes that parallel the fox squirrels and gray squirrels. And furthermore, this diversification happens early. This diversification of these 2 species of cancer cells happens very early in the progression process. For those of you that are interested, the cancer biologists, it was also associated with a variety of other standard markers. Ki67 is a proliferation marker, not surprisingly, on the edge of the tumor, where life is fast and dangerous. You have much more upregulation of cell division. In the interior of the tumor, where you may be more K-selected, there's less going on. Glucose uptake is much higher on the edge of the tumor than the interior of the tumor. HIF1 then is hypoxia. You see these upregulating the hypoxia, presumably in response to acidic conditions. And throughout, there was consistent patterns of differences. Now, this was at the whole scale of the tumor, going from the edge of the tumor into the interior of the tumor. But another feature of tumors is they have what's called angiogenesis. They have a vascularity to them that is somewhat supported by and created by the activities of the cancer cells. And any of us that have been down to the negative, been in Tucson in the Sonoran Desert, we know that we get these riparian habitats. And we know that along these washes that tend to then provide water, that what you get is a gradient of vegetation. What you see along the wash is mesquite, ironwoods densely packed, and then as soon as you move away then from the wash, we begin to see less densely packed vegetation, and furthermore, different species. Creosote bush, triangle leaf burr sage. So we ask the question, do you see the same pattern in tumors? These are blood vessels. Of course, this is three-dimensional, so you're cutting across them. And what we did is used a variety of stains, and what came out in all cases is that when you looked around the blood vessels, you found very densely packed cells, cancer cells. They're less densely packed away, much more densely packed here than away. So we see this pattern of vegetation. But to us, what was fascinating and I think significant is just like here, the species that are along the wash are not the species out here. As you can see from the stain, the cancer cells living near the blood vessel are not the same cell type as the ones living away. We see a very sharp break, and again, this pattern held up. And one place we looked for this then was in ER-positive and ER-negative breast cancer. Estrogen-positive cells are cancer cells that require estrogen from the blood to be able to complete survival and proliferation. Estrogen-negative cells are cells that have rerouted their metabolic pathways to be independent of estrogen. If you are a woman and you go to get diagnosed with breast cancer, what they will do is they will come up with the proportion of ER-positive and ER-negative cells, and from that they will make a diagnosis and prognosis. So for instance, up here, the purple cells are ER-negative, the green is ER-positive, these are normal cells, and again, in a breast cancer. And for this particular patient, 22% of the cells are ER-negative. This would be scored as ER-positive. The clinician or the oncologist will say that's a good prognosis, and then they will prescribe a course of action accordingly. This patient presents with 69.3% ER-negative cells. They will then be scored as ER-negative. The oncologist will say this is not good news, but we have a different battery of drugs. We have a different therapeutic regime. We will not give up on you. We're going to work with you. So we were interested in the question, why would you have such radically different frequencies of these two cell types within the tumor? To us, the obvious then is the amount of vascularity. We'd expect the ER positives to be the trees that would be living near the wash. We might find the ER negative is living farther away. And indeed, we looked at 24 women, and what you find is if you are ER negative, you have less than half as much vasculature within the tumor. If you're ER positive, you have over twice. There's a number of other reasons why there are therapeutic advantages and disadvantages in ER positive and ER negativity. But what you can see from this result is part of the bad prognosis from being ER negative may have nothing to do with the ER negative cells themselves. It may have to do everything with vascularity. The chemo agent, the hormonal therapy is delivered through the blood. If the tumor has poor vasculature, that means the drugs aren't getting to the cancer cells. So some of the negative prognosis or the poor prognosis of ER-negative breast cancers may simply have to do with the ability to deliver the drug into the right place. So when it comes to therapy, what does this mean? Therapy, I'm going to suggest, is a game of teleology versus teleonomy. Oh my goodness, what does that mean? So, teleology is design with forethought, designed by planning things out. Imagining, abstracting, and then designing. It represents anticipation. Teleonomy, on the other hand, is design of function through trial and error. Through trial and error. So anytime we tell our children, you know what? Don't do that. I have learned from experience. What am I really saying? It says, I really blew it at some point in my life. Boy, did I learn something by trial and error. So anytime you say, I've learned by experience, you have been teleonomic. Anytime you say, I thought this out, and this would be the smart thing to do, you're being teleologic. So here's the observation, what we're excited about. Wittingly or unwittingly, wittingly, as soon as the therapist, the oncologist, administers therapy, radiation therapy, targeted therapy, chemotherapy, immunotherapy, CAR T cell, take your flavor of the month. The moment therapy is administered, it is now game on. The therapist is now engaging in a predator-prey game with your cancer cells, and you are the playing field. And so the idea then is that the cancer cells, the good news is, they can never evolve to what hasn't happened. They can never anticipate what the oncologist is going to do. Thank goodness they cannot share information. You do not hear screaming little cancers as their patient dies, warning the patient in the next bed, warning the cancer cells, watch out, they're going to do CAR T therapy on you. And this is what didn't work. On the other hand, therapies can be teleologic, and in fact, they can be what we will refer to as a Stackelberg game, a leader-follower game. So we have a team of us, Maastricht University and at Moffitt, being led by Katerina Stankova, and that cancer therapy should be a leader-follower game. The oncologist should be the leader, and it should try to lead the cancer like a ring in a bowl. Why does this work like that? This is what sort of the models begin to look like as a leader-follower game. This is the resistance strategy of the cancer. This is the therapeutic strategy. And this is roughly how things proceed right now in therapy. This is where the cancer is. This is where the cancer would be in the absence of therapy. And then this line here describes what's called the best response. This describes what will be the evolutionary response of the cancer in time to all of these levels of treatment. So what happens is in precision medicine, we measure all of these wonderful things about the cancer as we should. We get genome profilings, we find out what mutations we can target in our therapy, and then we say this is exactly the right therapy that we should use for when the cancer is here. And then this is our best response. So we measure the cancer here, we apply this therapy, but if we do not kill them all, what is the cancer going to do? Evolve. So in time, the cancer evolves to this point up here. And then once the cancer is at this point, we call it progression. We reevaluate and we say, aha, this is the optimal second-line therapy. We then treat it like so, and then the cancer responds. Third line, we keep going. And eventually we arrive at the intersection of the two best response curves. That's called a Nash equilibrium. So when we engage the cancer by reacting to what the cancer does rather than anticipating what the cancer is going to do, we wind up at this Nash equilibrium. On the other hand, if we can anticipate that this is what the cancer is going to do in response to our therapy, we can look along this line and say, well, you know, this isn't so good. We really wish the cancer would just stay here, but it won't. So then what we can do is we can pick the point along this line that we feel is most favorable to the patient. This is called a Stackelberg solution. So we play this strategy down here, we just keep that strategy, and we can drive the cancer then to, at least for the patient, what is going to be the best outcome, whether it's a good outcome or not. What we do know from game theory, Stackelberg solutions are always at least as good as Nash solutions, usually a whole lot better. So what that means is we're aiming for evolutionarily enlightened management strategies. What we're aiming for then is appreciating, modeling, and anticipating the ecological and evolutionary consequences of management, conservation, and therapy efforts. This is well known from fisheries, antibiotic resistance, integrated pest management. None of this is particularly new. We know that our size-selective fishing causes large evolutionary changes. Right now, lobster fisheries in New England, Australia, in Canada are managed in an evolutionarily enlightened way in which they're protecting both the ecological stock and they're trying to protect the evolutionary integrity of the species. So there's much we can learn from integrated pest management. The first thing to learn is a pest is only a pest if it reaches density or engages in activities that cause undue economic, social, or health costs. This is well known, again, in pest management. Often best to manage rather than eradicate. We're going to come back to that one. Resistance will happen. Resistance will happen. And what we're interested in is we should have a resistance management plan. If you're being treated for cancer, literally look at your oncologist and say, what is your resistance management plan? And you'll get a big maw. And then you can go on explain. So what happens in typical new therapies? They will undergo a trial. They will come up with a new therapeutic, perhaps for metastatic ovarian cancer. They will then throw this drug at a large number of women, and then they will conclude that there is superiority. The women have extended progression-free survival for 5 months. The FDA in the United States will approve that drug, and it becomes standard of care. But never is it asked in the trial, why did it only give 5 months? Why did resistance occur in 5 months? Why couldn't it have been 10 months or 20 months? So what happens is we are always reacting, adding a new drug, a new therapeutic agent, and we are fatalistic. We accept the fact that it eventually will not work because that's just how cancer is. What's worse is we will often say the patient failed on therapy. The patient failed. Think about that for a moment. No, we failed. The patient did not fail. We failed. So the invitation here then is to anticipate resistance before it happens. Okay, so this comes into adaptive therapy. This was proposed by Bob Gatenby. Much of my work at Moffitt then is revolved around this idea of adaptive therapy, using models and measures of the past and present state of the individual patient's cancer to anticipate and steer the ecological and evolutionary dynamics of the disease by adjusting the time course of therapy accordingly. Right now, we do precision personalized medicine in which we define the appropriate therapy for where the cancer is. We then may do that therapy for 6 months, a year, 2 years, until the cancer progresses, and then we reevaluate. The argument here is maybe we should measure less in the beginning. We may be over-sampling and over-measuring initially, but that we should actually be tracking the course of the therapy continuously and then adjusting our therapies. So what I'd like is someday, I don't know whether you would find this gratifying or not, but to me personally, if my oncologist, when I have cancer, comes up and says, we are going to anticipate and steer the dynamics of your cancer, that will make me very happy. Yes, that as opposed to hearing what will now happen in many cancer centers, and it's not because they're not trying. The physicians obviously want to help the patient. They are dedicated health professionals. But what you will hear if you have metastatic pancreatic cancer that is inoperable, they will say, we will do our best, meaning they will treat to kill, maximum dose therapy, but we will not cure you. We will not cure you. But what they won't say then is because we cannot cure you, here is what we're going to try to do to steer and manage the therapy. So there's a variety of evolutionarily informed therapies that we identify. What I'll show you tonight is what's called competitive release therapy, double-bind therapy. Group sellout is the one that I would love to do. If any of you can help me develop a group sellout, this would make me happy if in the next 10 years. And then evolutionary gambit. What are these? Competitive release. Based on the principles of competitive release, the therapy moderates the evolution of resistance by maintaining sensitive cancer cell types that in turn compete with and suppress the more resistant type. The idea is you're playing ping pong. You can always treat the sensitive cells, but you back off. You make sure you leave enough sensitive cells around so that they become your therapy for the resistant cells. This requires that there be a high cost of resistance and that the sensitive cells have a competitive advantage. This is sort of the basic idea. If you do maximum tolerable dose, you're simply selecting, probably evolutionarily unwise, you're selecting for resistance, and eventually you have no therapeutic options. The idea that in adaptive therapy, you treat, you reduce the sensitive cells to an acceptable level, but then you let them grow back so that they suppress the resistant cells, and you continue this to maintain. So you then go for levels of therapy in which you maintain a livable, both for survivorship and quality of life. Does this work? Work done then at Moffitt showed in mice. Here is, if you implant a breast cancer in these mice, this is what happens to the tumor in these mice if you do no therapy. This is what happens under standard of care. And this is what happens if you simply treat the tumor when it becomes a certain size. All they did is when it got to a certain size, they administered therapy. And in fact, these mice were simply sacrificed at the end of 120 days because they had no progression whatsoever. These mice are getting exactly the same therapy as these mice. The only difference is the therapy is being modulated based on the size of the tumor. It's very popular right now to do metronomic therapy, drug holidays, but those are generally preset. They are not separate for each patient. In this sense, the tumor in the mice dictated the therapeutic strategy. Double-bind therapies, to use the use of 2 or more therapeutics, where the adaptive response of the cancer cell to one therapy makes them more susceptible to the second agent. All of you will recognize this as predator facilitation. As Bert and I know from the gerbils, the appropriate response for gerbils, resistance response to barn owl therapy, is to hide under bushes. Guess what clever snakes do? They hide under bushes. And so it turns out then the snakes, by being in bushes, owls being hunting more effectively in the open, owls make it easier for snakes to kill gerbils. Snakes make it easier for owls to kill gerbils. This would be an evolutionary double bind. I won't go into great details, but Antonia, he's one of the physicians at Moffitt, he was doing a P53 vaccine. The idea was to upregulate the immune response to then reduce the tumor. P53 vaccine was very, very effective initially. However, you got partial response, and then the cancer always came back. The clinical trial failed. But since these patients progressed, and he's an oncologist, he instituted second and third line therapy just to try to help the patients. These second and third line chemos have an expectation of less than 5% response rate. He was astonished to discover he was getting 60% complete or partial remission when using these second-line therapies in response to the first-line therapy. The suggestion here is this may have been a double-bind therapy. Group sellouts is one that we would particularly like to do. It's where the therapeutic approach causes the cancer cells to actually throw each other under the bus. How can this happen? You see it in the Far Side cartoon. Tail flagging and white-tailed deer, Allen's jackalope and antelope jackrabbits. What they're doing is they're telling the predator, I've seen you. They're not warning their neighbors. In fact, they're concealing it from their neighbors, and they're hoping the predator will reevaluate who it wants to kill. If you have resistance in a public goods game, it can be disastrous. So if the cancer cells benefit from the resistance of their neighbors, this beta is positive, then resistance is going to evolve rapidly and be very effective. However, with a group sellout, if beta is negative, meaning that the resistance strategies of my neighbors actually hurt me, and my resistance strategy helps me, what you can get is, as there's larger and larger neighborhood, the cancer cells will actually have runaway selection for their resistance strategy and actually create evolutionary suicide. Wouldn't that be wonderful? And that's the one, in the next 10 years, I want to create evolutionary suicide in a cancer cell. Can I have 5 more minutes, or? Okay. Thank you very much. Anyway, thank you for forbearing. This next therapy, which I think has a lot of promise, I can talk about it more, is an evolutionary gambit. This is a therapy becomes increasingly effective after the cancer cells and the tumor have been provided with a resource or good that by itself would actually promote tumor growth and more rapid lethality. But what happens then is the therapy, you're basically evolving the cancer cells into a corner by providing them a good. We can discuss this later, but now what I'd like to share with you is, can we do this? So we recently published this study, and this is what I'll be resulting on. So this was in November. We published then on a trial, a clinical trial, phase 1 trial on metastatic or castrate-resistant metastatic prostate cancer. This is a therapy or a point of cancer. This is a level of cancer no man ever wants. For the women in here, you do not want any of the men in your life, sibs, friends, partners, dads, and uncles, ever to reach this stage. This is the sort of the final stage of prostate cancer. And what you can see is all of these black areas represent metastatic tumors in the bone. The prostate cancer has gone to the bone. There can be anywhere from 20 to 25 secondary tumors spread throughout the femurs, the pelvis, even in the jaw, the scapula, the sternum. There can be 1,000 micrometastases. The last stage therapy is abiraterone therapy. In the response to abiraterone, 33% of patients will respond. The amount of response varies hugely among the patients, but eventually you will get progression, and progression comes in between 11 to 16 months. Within 11 to 16 months, there'll be some last-ditch therapy, and then these men will die. So every one of these men will die of this disease, and they will generally die within 2 years of this disease, or often much sooner. In analyzing these diseases, I'll talk more about it, we think there are actually 3 species. There's T+, these are cancer cells that require exogenous testosterone. There's this really nasty TP cell. These are actually cancer cells that evolve to produce their own testosterone, and they leak it out into the environment as a public good. The T- cells are cells that have become entirely independent of testosterone. So typically what happens to men when they are diagnosed with prostate cancer? They come in, they present with prostate cancer, usually and hopefully it has not metastasized to the bone. They will do local therapy. They've got a variety of therapies. And at that point, if those therapies are successful and there are no metastases, it will be curative. Prostate cancer can be cured. And it is cured then for those men where it has not metastasized to the bone. However, after therapy has been completed and they do not believe it's metastasized, they will track the PSA. This is all the PSA, which is used as a marker. And then for some of the men, this can be within months or within years, they will suddenly show a peak in PSA. And that alerts then the oncologist that the cancer is in the bone. And so this is cancer that was already there, small populations, these small undetectable populations, and at which point then they do androgen deprivation. They're all TP cells, and they will do what's called chemical castration. And at that point, you get a nice, long, deep response. This response can sometimes last for 3 months, sometimes for several years. They will keep them on Lupron, but invariably, if these men do not figure out how to die some other way, the cancer will become castrate resistance. And once it's castrate resistance, there's all sorts of different therapeutic agents that they will try, and eventually at the end, they will try abiraterone, is one of the last-ditch therapies. So this is what real patients look like and what real patients present. So here's an individual that underwent a radical prostatectomy, hopefully with the hope of cure. It did not. They were on Lupron. Notice they were on Lupron for almost 3 years. It begins to go up. They tried a variety of chemo agents. They finally tried abiraterone, and they got some success. This individual here presented then with metastatic cancer. They got androgen deprivation therapy. They got a response that lasted barely 7 months. Barely 7 months, at which point you can see the oncologist went for docetaxel, a chemo agent, fairly good response, tried abiraterone. Things look actually quite good for almost 300 days, and then death. The whole period then from presenting to this period here then is just a couple of years, and so on. So here's our hypotheses. Our hypothesis then is that metastatic prostate cancer has these 3 cell types. TP, you can identify the mutation. They provide a public good by contributing testosterone. T- are particularly inefficient and slow. They are very, very poor competitors, but you have no therapy for them. On the other hand, the T+ cells then require the exogenous testosterone. So like that blood vessel model, here's what we're imagining standard therapy does. This presents the normal cancer. You have then various T+ cells, very common near the blue ones are T+, even doesn't quite look like that. They're near the blood where they get testosterone. TP cells are a bit farther away, and the T- then are living in the desert far from the blood vessels. Eventually what happens is these cells will begin to grow and threaten to kill the patient, and then you do androgen deprivation therapy. The androgen deprivation therapy favors the TP cells, and now you apply chemo and abiraterone, and at that point the T- cells take over and result in death. The idea of the therapeutic strategy I'm going to show you tonight is that the men present with this. By the time they enter the trial, they've already undergone androgen deprivation therapy. So the idea then is to ping pong back and forth, apply abiraterone to knock down the T+ and TP cells, and then withdraw the abiraterone, and then cycle the tumor back to this stage. And the goal then is to create a durable, survivable cycle then of the cancer based on these 3 species. There are some challenges. Trying to find these 3 species can be a little bit challenging. We have stains for finding the T+. We have stains for finding the TP cells. Right now, there are no markers for the T- cells, so we have to use process of elimination. Whoever is not TP, T+, TP must be T-. But the goal is to find these individuals. So right now, we have then this pilot trial that we began about 3.5 years ago. The expectation is under standard of care, their PSA reaches a level, they go on continuous abiraterone, but eventually resistance. The idea here is, can we then keep ping-ponging back and forth and maintain a durable response? The underlying model, many of you will recognize. It's based on Lochner-Volterra competition models. Furthermore, it's a 3-species dynamical system. We generate then a payoff matrix based on the literature and the professional judgment of the oncologist. We then feed this into a 3-species Lochner-Volterra model and then into a model of expectations of PSA production. This is what model output looks like. Under standard of care, these are the different cell types. Under standard of care, we see the expectation then of a large response followed by resistance. This is what some of the model output looks like if you try to fluctuate the tumor burden within the patient. So the clinical trial right now is administer abiraterone until you knock down the PSA down to 50% of the level that was presented. Then at that point, you remove the therapy, and then you re-administer when the PCA recovers. The idea then is that each patient is setting their own benchmark. The benchmark is the patient's PSA. Our applying therapy and withdrawing therapy is entirely based on that patient. It has nothing to do with what's going on with the other patients. We have now enrolled 18 patients into the trial. We started just over 3.5 years ago, and this is what many of the patients look like. You can see this kind of cycling on and off. The first gratifying part is the clinical trial. The endpoint was that we would actually cycle. The endpoint was that we would get at least 2 cycles. If we did not get 2 cycles, in 2 of our first 5 patients, that was the end of the trial. All 18 patients that have been enrolled, they have cycled. In fact, we were really gratified to see that. We had no idea whether they would. The theory said they would, but we did not know whether they would or not. Every one of them has cycled. We have one gentleman who has cycled now 10 times, has undergone 10 therapeutic cycles. One of the things we've observed that's quite interesting is the model predicts that you can have slow cycles and you can have very fast cycles. And we see that in the patients. We have some of these slow cyclers where they can actually be on or off the drug for almost a year. Almost a year. And then over here you can see a very fast cycler going along in that particular patient. Finally, this is what's published in the paper. This is now back in November. Of the 12 patients that we can analyze, you can see on, off. Only one of our patients has actually progressed due to radiographic progression, but there is a caveat. This patient, after being on the abiraterone, and it was time to take them off the abiraterone, understandably, the patient was very reluctant to stop taking the drug that they thought was keeping them alive. So they pulled themselves off of the trial and wanted continuous abiraterone. They progressed in 2 months. In 2 months, that patient progressed. All of these other men have not progressed, and every one of them is still alive. This is the contemporaneous control cohort. We didn't have a true double-blind control, but using men that received similar therapy at Moffitt Hospital. Of this group of 16 contemporaneous control, at the time we wrote the paper, 14 of the men had progressed. 14. These 2 men have now progressed. There are none that has not progressed at this time. So as I give this talk now, of the 18 men, if every one of them were to progress tonight, if I got an email that said every one of these men has progressed, we are now at least 33 months. We have doubled what is the longest that's ever been seen in a study for standard of care. Furthermore, these men are receiving on average 47% of standard of care. They feel better. They're getting less drug. This drug in the United States is about $8,000 a month. And so they feel better. They're taking less drug on the trial. And so to conclude then with this particular trial, this is one of our patients, Robert Butler. Now, ordinarily you'd say you're not allowed to disclose patient confidentiality. I'm not going to tell you which one he is in those graphs or whatnot. But Robert Butler, which is fantastic, he's a trial patient. He then, after reading the paper, he's an engineer. Reading the paper, he wanted to understand the mathematical model that is keeping him alive. So he has now become a patient advocate, and he's become a friend. He's been on trial for 3.5 years. The other two men that were on contemporaneous treatment, sadly, they're dead. So hopefully, rather than being the end, we hope this is the beginning. Thank you very much.